Hi, I'm Spoon, and in this video, I'm going to talk about this 2019 article titled Hydrogen Peroxide Gates a Voltage-Dependent Cation Current in Aplasia Neuroendocrine Cells. For this video, I'm going to partly talk about this paper, but I'm also going to use it as an example of what science looks like. Before I started my bachelor's degree in bioengineering, I had a limited idea of what research looks like. And I think it would be useful for students who are going to do research in university to have an idea of knowing what it might look like, as well as just give an insight to people who are curious about what does the nitty-gritty of science look like. So without further ado, let me talk about this paper. 3 years ago, I was taking a course called Biology of the Brain. And one of the assignments for this course was I needed to pick a paper in neuroscience, read it, understand it, and make a poster presentation. This poster presentation is supposed to be a practice in scientific presentation and speaking, and also proof that I'm able to read something and understand it. That was 3 years ago. It has been a while. But I still really like this paper because some of the things that it found were rather interesting. So, about the science aspect, let's make up a scenario. Let's say you are in high school and you think the human brain is very interesting. You've learned a little bit about it, and you decide that you want to study biology or neuroscience and eventually have a career as a scientist where you are understanding brain diseases and how to make them less of a problem, either in detecting them early on in early stages or finding different therapies and treatments to cure people, alleviate symptoms, something along those lines. You just know you want to do research on the human brain. So you enter university and you start your degree in biology, and eventually you learn a little and find a laboratory at your university that does brain research. You reach out to the principal investigator of this lab. You say you want to be a part of their lab and do some research yourself. If you're lucky, this principal investigator will have money to hire you depending on your level of experience, or you might end up volunteering, which I think it's really good to do research and make sure you're getting paid for it so that that can be your livelihood. Otherwise, it can negatively impact your performance in coursework and such. But it's also good to have experience, and working in a lab can be very, very educational and add to what you're learning in college. Anyway, let's say one way or another, paid or not, you are able to start working on a project in this lab. The project has already been decided. It's going to be about hydrogen peroxide and what it does in the brain. And let's say your job is to look at a bunch of different prior research and create a paper on it. Um, a little bit about that. In the process of research, early on starts with a question, and someone comes up with a research proposal, finds some ways for the research to be funded because it always costs money to do research, unless it's a literature review in which you're just looking at a lot of different papers that is a little bit less expensive. But in any case, there's some work behind the scenes that can happen before research even begins to start. So let's get back to this. You're looking at a lot of different papers, and one of them you come across is this one, hydrogen peroxide gates a voltage-dependent cation current in aplasia neuroendocrine cells. So let's get into it. This is a primary research article, which means that these authors right here did their own background research. Maybe they started writing this paper before experimenting, but they did their own experiments to reach results that haven't been found before. They're doing their own firsthand experimentation. And primary research, most research articles, I hope, have this thing called an abstract. This is for people who are looking through a bunch of different articles we'll look at before deciding if they want to keep reading, because papers take time to read and you want to know if this is the right paper you're reading. Now, the search might actually look like this. There are different places that you can look for scientific articles like journals like Micro Machines, IEEE. Um, but let's just look at PubMed, which is a government database of a lot of different articles. So um, I'm looking up this paper specifically, but what if I looked up hydrogen peroxide brain, whatever, and maybe this will pop up. And the nice thing is, um, when you find this, you get the title, author information, identification numbers. Then you have the abstract here, which is the summary of the entire paper. And fortunately, this is open access. You don't have to pay money to read it. Um, if you are affiliated with a university or your company has a subscription, then maybe you're able to access it, but it does cost money. So here I can just access the article. Hopefully, I'm showing you now that if you want to find this article yourself, this is how you can access it. And here is the entire thing. After the abstract, the paper truly begins with the introduction. This gives some background information and then starts getting into a research question. And then towards the end, it summarizes what the experiment was that was done and some of the findings and conclusions reached. Then after typically is the materials and methods section. These are the procedures that are used to do the experimentation, what equipment, what materials. Um, it also might mention uh, ethical considerations, like, for instance, an article may say, um, this was all done in accordance with an institutional review board that makes sure that an experiment done is ethically, especially if there are live specimens or human subjects. Some papers, um, the materials and methods section are at the bottom. Every paper can be different. Sometimes sections are combined. But typically after materials and methods are the results section, which goes into what are the findings. The results section sometimes is combined with a discussion section, but when on its own, typically there are no interpretations. The authors are just giving the numbers that were collected or whatever results. There's no analysis or limited analysis. And then in the discussion, it talks about the significance of the results. This has a lot of results. There we go. Um, so it talks about in the discussion section, the authors might go talk about what was already discussed in the introduction, get into the significance of the results they found, and then arrive at some conclusions. Sometimes, if you're lucky, the paper will have its own designated conclusion section. Um, and then towards the end, there's maybe an appendix and a bunch of references. All right, so you saw how this paper is organized. At the very top is the abstract to let you know a summary of the paper. Then you get into the introduction, materials, methods, the results, and the discussion. And I think that's as much as I want to get into looking at this paper itself because it is very complicated. And 3 years ago, when I was reading this, um, took a lot of rereading and looking up a lot of different things. For instance, um, I already knew what, like, for instance, tetrodotoxin is, but I didn't know what this is, which is abbreviated as NAC. So in any case, I don't want to force you to have to learn every single thing about this paper because that will make this a very, very long video. And I just want to focus on the science bits. So here is the poster presentation. If I haven't already mentioned, this was a great poster for the class. I did what I needed to do to get a grade, a good grade for this assignment. But if I were to be in a conference or a symposium giving a poster, this would not be a very good poster to have. It's good to have a poster with a lot more images and graphs and figures so that someone can look at your poster, quickly get an idea of what's going on and seeing some important results, and any of the more finer details I can just explain orally. But this gives a much more uh, simplified version of this article. Not everything that is discussed in this article will appear in this presentation here, but I think it gives a much more, I think it follows the most important narrative that is being given. So let's talk less about science in general and this paper in particular. So summarizing the introduction, hydrogen peroxide is generally considered a reactive oxygen species that is toxic to cells. Hydrogen peroxide you can buy. Uh, Peroxide. You can get this at the dollar store, um, or imagine at a pharmacy. It's often used for first aid. When you buy it, typically it is a very dilute solution of hydrogen peroxide. So it's mostly water. And that is because in first aid, it can be used to disinfect a wound, but it is also particularly toxic and dangerous in higher concentrations. Um, so it can kill bacteria in a wound to make sure you don't get an infection. Although I've heard of some controversy whether this is more damaging than beneficial. You'll have to look that up yourself. Um, but you can get this as a first aid uh, solution. Where's my PowerPoint presentation? But overall, before getting into finding this article myself, that's what I was taught. Hydrogen peroxide is just toxic. It's naturally produced due to different metabolic activities in humans and other species. Um, but in general, I was thinking, oh, it's just good to get rid of because it's toxic. However, this paper shows that not only the results that they were found, but also prior research that was mentioned in the introduction of this paper. There's research showing that it may have a role in ion channel regulation and cell signaling, which means maybe there is a functional purpose of hydrogen peroxide in biological processes that does not consist of destroying things. So, in this particular study, bag cells from the species Aplysia californica are studied, and these are neuroendocrine cells that oversee egg laying hormone secretion. All right, so Aplysia californica is a sea slug. It is, if not my favorite, one of my favorite animals. I have had to learn a lot about this sea slug. In neuroscience, it is used as a model organism. Let me see if I can find, here we go. Aplysia californica has very large neurons and a rather simple nervous system compared to humans. Right, this right here is a diagram of its brain, which is divided up into various ganglia. So this is often used for neuroscience research, and this is used as a simplified model of the human nervous system. So here are our neurons that we'll be studying. And then in the study, it focuses on bag cells, which are important for egg laying. These sea slugs reproduce by laying eggs. And so to summarize what this studying is doing, it is seeing, does hydrogen peroxide affect the signaling done by bag cells that control egg laying, which would imply, what if hydrogen peroxide is important as a cell signaling molecule that controls the process of egg laying? Which is very interesting because that would imply hydrogen peroxide is not just toxic. There's already prior research, but this is focusing mainly on the egg laying aspect. Now, hydrogen peroxide um, naturally is produced through the cell signaling processes of these bag cells. So it's naturally present. What if it's not just toxic? So the introduction kind of goes into why Aplysia californica is used. So in the materials and methods, the authors talk about how specimens are gathered. I imagine they had to confer with an institutional review board to make sure that their handling of these live specimens are done in an ethical manner or as ethically and humanely as possible. And then in the materials and methods section, um, the authors talk about what equipment they used, what chemicals they use, and how they are testing, does hydrogen peroxide affect cell signaling? A lot of different factors are controlled, but I think the most important thing to consider is that they're measuring how cell signaling is affected by the presence of different concentrations of hydrogen peroxide. Either they add hydrogen peroxide or they control the amount by adding different chemicals that add or remove hydrogen peroxide. And to summarize these results, it looks like that higher concentrations of hydrogen peroxide increases the cell signaling activity of these bag cells. And so looking to the conclusion, the study supports that extracellular hydrogen peroxide induces an inward current at a rate that's dependent on the. All right, summarize, hydrogen peroxide does have effect on, it's suggested based on these results to affect cell signaling, and it may have a role in egg-laying behavior in Aplysia californica. Now, this paper seems to mainly focus on that its relevance is based on challenging the stereotype that hydrogen peroxide is completely toxic. I would like to build off of that. As a bioengineer, let's say I consider hydrogen peroxide as strictly toxic. And let's say because hydrogen peroxide can damage DNA and maybe that causes cancer, I decide, what if it would be beneficial to look into a cancer prevention drug that reduces the presence of naturally occurring hydrogen peroxide in the human body? Which sounds great, but if hydrogen peroxide is important for different processes, maybe that would also be damaging. So this study is interesting in challenging the narrative, and I think it does have some important real world implications and future research on things that relate to hydrogen peroxide. To add on that, I'm currently studying health physics, and the textbook I'm reading introduced the theory of hormesis. Uh, this is how it's spelled. According to the textbook, which I believe was published about 20 years ago, so maybe this is old information, there have been studies that showed a potential link between how elevated natural radiation, background radiation, could correspond with lower cancer rates. Radiation, ionizing radiation can be very good at producing hydrogen peroxide, which is toxic. So high amounts of radiation can produce toxic amounts of hydrogen peroxide, but it seems like a certain amount of elevated radiation could be beneficial, which would imply more hydrogen peroxide. So gets into this nuanced potential of what hydrogen peroxide could have as a benefit. So maybe if I were to produce a drug that um, reduced the amount of hydrogen peroxide excessively in the human body, maybe that ends up actually increasing cancer. That is just a theory, though. And I think that's an interesting scientific question that needs to be tested before it's taken any seriously than what I'm saying right now. All right. And so we've read this article. Now, for these authors, they did this experiment and then they have to submit it to a journal and they might have to pay the journal in order to start the publication process of this paper. Then some reviewers, I think often volunteers, look at their paper and give a lot of comments, and the authors might have to adjust some of their claims or rewrite certain bits, hopefully not being told this experiment is flawed, you need to do it over. Um, but in the end, after different experts in the field look at their paper, then it gets published. And now I'm reading it now. Um, and I have not mentioned too much how complicated this is, at least in my opinion. There's so many different factors that were adjusted in this experiment to test like different salt concentrations, different voltage potentials or electric potentials across the membrane, how different pharmacological agents that either affect the hydrogen peroxide in solution versus affecting the cells being tested directly, how did that impact? But yeah, if you're going to do research, you're going to have to read papers that are very confusing, have to teach yourself new things to better understand what is going on in these papers. And yeah, so far I hope you're getting an idea of different considerations that need to be taken while doing research. What does it look like having to look at a paper that may not be very complicated, trying to understand why different methods were used, um, and extracting what is the relevance of a paper. And I hope you found this paper interesting overall, because I think it's very interesting in how I overall was taught hydrogen peroxide is generally toxic, but maybe it's not completely. One last thing I would like to mention is statistical analysis. Interpreting results can be very subjective because let's say um, we're looking at this graph. This graph shows different points, data points that were collected. The x-axis shows concentration of hydrogen peroxide, and this is showing the electric current passing through a cell membrane, which is corresponding with cell signaling. So to look at this graph, this curve, there's a curve that is being drawn through these points, and it looks like it is going upward, saying that increasing levels of hydrogen peroxide corresponds to increasing cell signaling. One scientist may look at this and say, hey, this looks like um, there is a strong correlation. But another scientist may say, how do we know this? Do we have enough data points? Is this a large enough difference that this probably didn't happen by random chance? And that is where statistical analysis comes in. It is a mathematical method that considers different assumptions about the nature of the data, how much data there is, and does the data show enough based on a few assumptions that there is a statistical significance. Ultimately, the criterion of statistical significance is dependent on a single number called a p-value. And generally, the convention is if the p-value is less than 0.05, then the results are considered statistically significant. I won't go too much into that, but um, yeah, science needs to make sure it's not relying too much on subjective opinion, but rather on specific quantifiable criteria um, in order to avoid things being solely based on opinions. I lied. I said the statistics part was the last thing I want to talk about. The very last thing to end this video, for an experiment to be mean anything or worth anything, it has to let the opportunity be possible for the authors to look wrong. The authors tested a hypothesis about whether or not hydrogen peroxide does in fact have a potential role as a cell signaling molecule. In order for this experiment to mean anything, there has to be the hypothetical situation that data is found that completely contradicts what they're suggesting. So let's say maybe that looks like this curve that is just completely flat. It doesn't look like any change in hydrogen peroxide concentration leads to any difference in cell signaling. That means their hypothesis is falsifiable. And if you think about it, if you have a scientific question you are testing, and there is no possible data that can contradict you, or no data that you will accept that will prove you wrong, there is no point in doing the experiment because you never had the opportunity to be proven wrong. Whatever results you got wasn't going to um, change the answer you were going to reach. All right, so this was a lot of me talking about science, this particular paper, and yeah, in the situation, you are looking at this paper as a part of hydrogen peroxide research because you have an interest in studying the human brain and helping with treating diseases. This would be one of hopefully many, 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 many papers that you will have to look at to complete your review paper. Um, yeah, in science, it's really important that you're able to test something, repeatedly test it, and get consistent results. That way you can show that what you're finding is not random and it supports whatever claim. Alrighty, I hope you enjoyed this video, maybe learned a little bit more about what does it look like to do science, what does it look like to think like a scientist, and also this specific paper on neuroscience, knowing some things about neuroscience and what the significance of this paper is. And I hope you have a great day. I gotta stop recording.